So today we'll be talking about drop in Cytobank. If you're unfamiliar with drop, however, and Cytobank in general, I'll just take a second to walk you through it. There are three main components of Cytobank. The first two are part of our cloud-based software. The first one is our high-dimensional analysis tools. These are things like Visney, Spade, and Citrus. I'm sure by this point you've heard about at least a few of those. In addition to that, though, we also have structured content management. And these are tools that help you to collaborate, organize, and archive your data, all in the cloud. So outside of our software offering, though, we also have an informatics consulting group. So at Cytobank, this is a group that does bespoke analysis. So if you have a large data set or planning a large clinical trial, please get in touch with us. If you'd like help with that, let us know, and we can talk about it. But today, for this webinar, I'll be talking mostly about drop. So what is drop in Cytobank? Drop is a new feature that we've just released on all of our enterprise servers that allows you to upload tabular data and then analyze it with traditional cytometry analysis tools, such as gating and heat maps, but also our advanced algorithms like Visney, Spade, and Citrus. So where can data for this come from? The obvious answer is things like single-cell data. So if you have single-cell RNA-seq, or if you have segmented image files, immunofluorescence, or immunohistochemistry images, or data off of Fluidigm's new mass cytometry imaging instrument, the Hyperion, all of these, once they're segmented for the images, will work. However, you can also look at bulk data. So these are things like Luminex or mesoscale discovery data, things off of the nanostring, which makes sense, but you can get a little more creative. You can tack on things like clinical outcomes. So if you have pain scores or just annotation channels about if a patient is surviving still, or if they're old or young, you can add that data into sample-based tables. You can also look at metabolomics data or bulk next-gen sequencing. But then if you want to get really creative, you can put in non-science data. So if you wanted to look at website stats or marketing data, or if you go on Yelp and pull some data down and look at restaurant reviews, all of that data, as long as it's kind of conceptually organized, as in each row is an event and each column is a channel or an attribute, you can put that into Cytobank and then run our traditional and advanced tools on that data. So in this webinar, I'll be covering kind of three examples of how we've used drop so far, and it's kind of a jumping off point for you to then use drop. And I'll have some slides later on talking about how to get access if you don't currently have it. So the first one is a data set from the TCGA. It's several kinds of kidney cancer and some normal controls, and that's bulk RNA-seq data. The next thing I'll be talking about is a data set we have from NanoString, actually. It's their new 3D biology platform, and that has bulk or kind of sample-based data on genomic RNAs, SNVs, and indels, but also protein, total protein and phosphoprotein, all in one platform. So I'll look at that. And then lastly, we also have a collaboration with the FDA, and that's on segmented image data. So the first data set that I'll be talking about is, like I mentioned, it comes from the Cancer Genome Atlas. It's a large database online of many kinds of samples from many kinds of cancers. We've taken ones just from kidney cancers, and we are basically taking a look at, I think it's 5 or 6 different kinds, plus some normal controls. So what can we learn by running Visney on this kind of data? Obviously, the hope is to be able to separate them objectively without having to, kind of manually get in there and organize where these samples come from. The goal is to have the algorithm do it for us, and without kind of interference from humans. Again, that's where the objective comes from. So this data set, if you'd like to try it yourself, is available on all Cytobank servers. So if you have access to a Cytobank server, if you have an account, you'll be able to poke around with this data set and experiment, kind of reproduce these analyses that I'm going to show you in this next little bit. Just a note, and again, I mentioned this before, drop is available currently on enterprise servers and not on premium, but this data set is. So if you'd like to see and interact with it a little bit, that's a great place to get started. So what does this data look like? The values themselves from the experimental channels are in FPKMs, which is fragments per kilobase of transcripts per million mapped reads. And this is obviously a little bit different if you're not used to looking at genomics data. It's not like MFI, but it's a standard way to represent RNA-seq data. In addition to these experimental channels, though, we also have some annotation channels. So these are listed in this particular spreadsheet, and they're at the very end. And it's we've actually converted them to integers, but it goes by gender, vitals, age, disease type, tumor stage, and sample type. So these are kind of some parameters we can use as a key to look at how VISNI has done to separate our data. And as a note, normally if you're dealing with genomics data, you're looking at thousands or tens of thousands of channels, as it were. In this case, Cytobank right now has as an upper limit on the number of channels at 816. So if you have more than 816 channels in your tabular data, it's a good idea to reduce those to be under that benchmark there. And you can do this a couple different ways. For this particular data set, we went through and just sorted by variance. And what that got us is I think it's 806 most variable genes in this data set. And then we use those in our advanced analyses. However, if you would like to use a different method, like PCA, principal component analysis, that's a very viable way to reduce the number of channels in your data set to just the ones that contribute most to the change between your samples. However, if you do that, you might not be including channels that you care about. For instance, if you care about Tregs or immunotherapy, you might really be interested in looking at PD1 or FOXP3 or these other things. And they might not change the most. It's totally okay to tack those on based on your biological question. So if you don't have a highly variable but still very interesting gene or protein or clinical outcome, again, by all means, include that in the data set. It just has to come in under 816 total channels. So what does this data look like? The VISNI combined with coloring by channel allows us to look at the expression levels of different RNAs across these channels or across these samples. So here we can see that this island, VISNI has separated our kidney cancer samples into these 5 kind of main islands. This island is high in IGF-2. And if we look over here, this island is high in VEGF-A. And this is useful. You can kind of see, I guess these contribute to the reason why VISNI has separated these islands. However, if you then take a look at our annotation channels, this makes a little more sense. So here we're coloring these islands by, or these samples by the type of cancer the patient had, and the type of cancer, hopefully, that was in this sample. And you can see it kind of groups into these 5 main regions. And this rhabdoid tumor and normal solid tissue, it's actually normal tissue from patients with a rhabdoid tumor. So that's why they're kind of close together down here. But it's done a pretty good job of grouping by tumor type. And if we go back and reference the previous slide, this high-risk Wilms tumor is high in IGF-2. And if you go into literature and look, there's a good record of Wilms tumors expressing or overexpressing IGF-2. And same with renal clear cell carcinoma. There's a clear record of that kind of cancer overexpressing VEGF. So it's nice to be able to kind of draw these direct connections between the samples we see and what's been established in the literature. Another thing you can do if you have more annotation channels, like for instance in this data set, this is just one of them. What I showed you before, there were probably 6 or 7 different annotation channels. And if we look at the age distribution across our different islands or cancer types, you can see that really only the Wilms tumor has patients that have been under 20 years old, and that's again that's just how that cancer operates. It shows up sometimes in people under 20, and the rest of these are usually acquired later on in life. So this is another nice way to kind of visualize the data. And this coloring mode in Cytobank, if you're familiar, is it's a dot plot color by overlaid figure dimension. And then we've gated out these cancer types or these age groups as figure dimensions. So the summary of this data set is that it turns out that using drop to convert this data to an FCS file and running VISNI on it has definitely allowed for an effective visualization of these experimental and annotation parameters. So we can see, yeah, sure enough, it did separate these cancer types. However, I should take a second to point out this is where it gets really interesting. You can see there's some blue ones up here, and there's some orange ones down here, orange ones in this big blue thing. And what are those about? Is that like, you can't say it's doublets because this isn't, this is bulk data. It's not cytometry data. You can't, use those typical explanations anymore. And these are biopsies from patients. And so if there is for instance, an event that is classified as a tumor, but it clusters right next to all the normal controls, it was a missed biopsy. They didn't actually get the cancer or the tumor in the biopsy sample. They may have gotten some normal tissue from around the cancer site. How about, if they're in this kind of cancer, groups of this kind, maybe it's a misdiagnosed type of cancer. Obviously you need a lot more information to actually make that call, but it's a thread to pull on. So this kind of visualization allows you to see what kind of a traditional analysis blinds you to. If you go through thinking, oh, well, I got this sample from this clinic and they said it's for sure this kind of cancer, you kind of take that for granted. But if you look at it in the context of what it actually matches with compared to the rest of this data set, it's a really interesting, like I said, idea and something to investigate. So the next data set that I'll be talking about is from NanoString. It's part of their 3D biology platform. And the previous data set we just looked at, the TCGA one, is just RNA-seq data. It's bulk RNA-seq data. This NanoString 3D biology technology allows you to look at up to 800 single nucleotide variants, RNA, proteins, and phosphoproteins, all in the same sample at the same time. So in this particular data set that we're looking at, there ends up being 104 SNVs and small indels, 192 different RNAs, and 28 total in phosphoproteins. We're looking at those in 144 different samples from 3 different melanoma cell lines. So so you can wrap your head around kind of what we're going to be looking at. So NanoString 3D biology, essentially, DROP allows you to take this spreadsheet data that NanoString generates, and it converts it into something that Cytobank can handle. And so here we're looking at these different RNAs and these different proteins, and you can see, just like the other data set, differential expression across these different islands. And when you look at the key, again, this annotation parameter, you can see that VISNI has separated these islands into their different sample types, or in this case, technically, the different genotypes: wild type, homozygous, and heterozygous for BRAF. In this case, VISNI again has allowed us to look and run, or DROP has allowed us to run VISNI on non-cytometry data to look at these differences across these different samples. But we can take it a step further. VISNI is a great tool. A lot of people use it, in kind of an introductory way. But this is something that you can riff on in Cytobank. So there are the VISNI results, you can then link SPADE and VISNI together. So you can run SPADE on the VISNI results and get hierarchical clustering on these t-SNE results. So this basically objectively clusters these groups. There's no kind of manual gating involved, and now we have each of these kind of subpopulations inside of these larger samples grouped for us for further investigation. So you can get a little fancy, and you can, just like other kinds of data in Cytobank, you can link our algorithms together. Not to say that new is always better. You could always go and look at this other kind of data, whatever other kind of data you've put into drop, in our traditional cytometry display methods too. So normal heat maps or histogram overlays. All of the functions of Cytobank work on this drop data. So the summary of this is basically just like the other one. We've allowed these unsupervised machine learning algorithms to look at hundreds of different markers and for the effective visualization of the differences between our samples. So that's nice. And here for this nanostring data set, we've looked at genetic data and also protein data. However, it is possible to take this a little bit further. So if you have sample-based data, and I've mentioned this before, and you have clinical outcomes on something. So maybe you have a patient sample, so you know attributes about that patient, and you take that and run it through a sorter, and then run a nanostring on it. You can look at outcomes and link that data in a spreadsheet before you upload it to Cytobank. You can link that data in with the protein and the genetic analyses. So you can have and group on and run VISNI on outcomes and genes and proteins all in the same file. So it's a very effective way to do that. And even if you don't have numeric values, so for the outcomes, like if you have a pain score that's numeric, and that might be something you want to cluster on. But if you have literally just healthy and disease or healthy tumor, any other kind of thing, you can turn that into an integer, and it would not necessarily be appropriate to cluster on it, but you can still use it as an annotation channel, like I've shown previously, to help kind of understand what's going on in your data. So this is very extensible. It's not just using one data set. You can kind of the sky's the limit. Obviously, you have to be intentional about what you're linking together and make sure it references the same sample. But if you have questions about whether that might be appropriate or not, by all means, you can write in and let us know. And the last data set I'll be talking about is we have a collaboration with the FDA to identify functional subpopulations based on morphology that could be selected or enriched for these immunosuppressive therapies that everybody's dabbling in. It's definitely the hot topic these days. So to do this, and I mentioned this before, but once you have segmented data, Cytobank is a secondary or tertiary analysis platform when it comes to image data. We don't do segmenting. There's a lot of other people out there who are really good at it, using lots of different software packages, and that's not something we're getting into. So once you have segmented data, you can upload it to Cytobank and then run our algorithms on it. And that's what we're doing here with the FDA. So it's still kind of in process, so I don't have any visible data to show you, but keep an eye out. We're releasing stuff as it becomes available. But essentially, the workflow for looking at segmented image data is do the segmenting, and then once you have both the protein and the morphology features in a tabular data format, you can then, using drop in Cytobank, convert that and then run our advanced analyses, or our traditional ones like gating or heat maps, on both the proteins and the morphological features of those cells. So this is definitely something that I think people will be interested in. It's a kind of a new take. People have been running things like t-SNE on genetic data for a while. But this is where things are starting to develop in the imaging field. So kind of in summary, why would you run cytobanks analyses on other data types outside of cytometry? And the answer is to compare these samples in an objective manner across many dimensions and display the results simply and effectively. These tools are very simple to use, especially if you're already used to using Cytobank. That's the goal. That's why we put them in our software, is to give people who don't have access to a computational biologist or bioinformatician access to these powerful bioinformatics tools. So we're now opening up the doors to these other data types, so you can use these algorithms on other data that you work with, and it can also be used on existing data. So this, I think, is a valuable point to mention, is you don't have to say, oh well, you can run it on only new data. If you have 5-color data from your imaging core 10 years ago, you can then go back, look at that data, convert it, and run Visney on it in Cytobank. So definitely the sky's the limit when it comes to how you can apply this. And hopefully today has given you a little taste of how that might work. So I'm going to hop in after this is done and kind of show you a little bit about how it works actually in the software. If you have any questions, feel free to write them into the chat window, and I'll have an address coming up in just a second. But, and I've mentioned this before, how do you get access to this? So there's kind of 2 main versions of Cytobank. Premium is the a la carte version. Enterprise is a named version of our server. It's for your site. And it has some distinguishing characteristics. And for right now, Drop is only available on our enterprise servers. If you'd like to try Drop and you don't have access to a Cytobank enterprise server, please get in touch with us to talk about how you can get access, because we're willing to chat. So if you need access to Cytobank, if you tried it a long time ago and you made an account and you're like, oh, you know what, this is cool, but I used my 30-day trial, that's totally okay. Write into us and we can give you another extension. If you haven't tried yet, please go to premium.cytobank.org and try Cytobank free for 30 days. And it is the summer, so if you're on vacation and you do this and you forget about it for a month, and then you're like, all right, it's September, I'm going to get back in the game, and then your trial's extended or expired, let us know and we can extend it. But for more information on Cytobank or on Drop, please go to support.cytobank.org and look it up. We have a support article there with lots of different kind of examples, and we'll have kind of example video up on YouTube. And as always, other questions, please feel free to write into sales@cytobank.org. So I'll take just a second and answer some of these questions that people come in. Let's see. All right, so people have asked, hey, is it possible to get in touch to talk about whether this might be useful for your own data? Absolutely. We're happy to set up one-on-one WebExes or Google Hangouts to chat about it. Just again, write in at support.cytobank.org. Another person asks, what kind of data types? That's a good point. I am remiss if I haven't mentioned that I say tabular data types right now. We're talking about .csv or .tsv files. These are comma separated or tab separated values. This is kind of the PDF of the number world. A lot of other data types can be converted into a CSV, and then you can take that and upload it to Cytobank. So this is our way of opening up the doors and making it easy for lots of different kinds of data to come into Cytobank. But we don't work natively yet with any image formats or genetic information formats. Let's see. It looks like that's all the time we have for questions. If there's a handful of questions here that are more specific, and I'll get back to you specifically about that. Oh, here's a few more. how many rows do you need to run Visney and get useful data? So that's another very good question. So in Cytobank, to run Visney, you actually need, if we're being technical about it, 3 times your perplexity. So perplexity is a value you can change in Cytobank, and if you have, 60 events, it's best if you have 180 different events, and that could be samples or 180 single cells from a segmented image or tissue section. The enforced minimum is 60 events, and again, you can play with the perplexity to get that to work out. But that's what you should do. So if you have 15 samples, unfortunately, that's not enough to upload and run Visney on in Cytobank. So either try adding more, or just copying and pasting those files and then uploading 30, or doing it again to upload 60. That's not okay. Statistics tells us that's inappropriate, so I can't recommend that. So can column A and B be gene ensemble and gene ID? So that's actually a good question. I'll actually hop into the actual data table to show you what the TCGA data looks like. So in Cytobank, when you upload a spreadsheet, you can choose kind of which cell is the beginning of your data section, of your numbers. So in this case, I would select this cell in their interface, and I can show you that in just a second. But you'd select this cell, and that would mean that the first row is the annotation or the channel names, and this is the first column. So this is the first channel we're dealing with. So column A and B, you can have columns in Cytobank that are non-numeric when you upload it. However, you'll have to leave those out of the data. So we end up converting all subsequent columns into this FCS format, and that ends up kind of throwing a wrench in the works if you have text in one of the columns. So you can either hide that column, or you can convert it to a value and just say, this is my gene ID _ annotation channel, and so not to cluster on your gene ID or cluster on the ensemble. But yeah, there's lots of different ways of kind of tweaking your data to get it to work. So one last question. So you can access Drop with the 30-day trial of premium. That's kind of the point I was getting at. So right now, Drop is not active on our premium server. So if you have a 30-day trial or would like to start one and get access to Drop, write us in, and we have kind of a beta server off to the side that you can potentially poke around in. So please let us know if you'd like to, and we can take it from there. For those of you who have more pointed or specific questions, we can follow up after this webinar by all means. So with that, I'll actually hop in and just briefly show you what this looks like. So if I wanted to make a new experiment, I'm actually going to, it's just like normal. You give it a name and a purpose, and then click Create Experiment. And I'll come over here. And let's see, we are going to save this. And we're just going to drag and drop it. So let me find this on my computer real quick. And drag and drop. And this is that CSV file we were just looking at. And there's a little option down here that says convert all CSV files to analysis files, which I'd like to do. Now click upload. And then just for our metrics, just so we can know what people are using, we ask that you say, hey, this is RNA data or DNA data. In this case, it's RNA bulk data. So I'll click that, and I'll click submit. And here is where you get to choose, like I mentioned, which columns start your actual values. So if you have two kind of annotation columns at the beginning and you don't want to use those, you just leave them out. You can click on this cell, and it'll say, all right, the first row then is all of my annotations. I'm skipping column 1 and 2, and column 3 is the beginning of my real data. So you get to select what you want. You can actually list a series of columns as well to skip if you know, for instance, that column number 16 is something you don't care about. But once you click on that, you confirm that that is indeed how your data matrix looks like. It'll process the files and then give you that now as data inside of Bank. So once it's converted, you can then start gating on it. You can start a VISNI, a citrus, or a spade, and take it away. So that's the workflow. The goal is to make it as easy as possible. If you have questions about the specifics about what can and can't be in the FCS or the CSV file or TSV file, that's listed on our support site. If you don't have, if you've never been there before, I can certainly point you that way. It's support.cytobank.org, or in the software you can click on this support home little life preserver, and that'll bring you straight there. And you can either look up our knowledge base or write us an email from that point about questions you have. With that, I think I'll call it a webinar. But thank you very much for your time and attention. And like I said, check out Drop if you have a chance. Thanks.